Hi everyone, we're progressing on to the cardiovascular system. The cardiovascular system is a pretty big unit. We'll start with the heart, and then we'll talk about the vessels and also the physics behind how blood circulates through those vessels. So lots to talk about, so let's get after it. Uh, where is the heart located? It's located in this region that's kind of in the thoracic cavity, surrounded by the two sets of lungs. So in your thoracic cavity, it's usually divided into four compartments. There's what's called the right pleural cavity, the left pleural cavity, and each one of those cavities contains a lobe of the lungs. There's this region here called the mediastinum, and then there's a superior portion of the mediastinum and an inferior portion of the mediastinum, and then embedded in the inferior portion is this special cavity that we call the pericardial cavity. So this isolated spot that houses the heart. It's a great place for the heart, though, because the heart has really two jobs. One to send blood to the lungs and back, and one to send blood to the rest of the body and back. And so it's, a good design if one of those jobs can be completely minimized so that no extra energy is wasted. So this position next to the lungs is kind of strategic. It minimizes the energy required to do at least one of the jobs that the heart does, sending blood to the lungs and back. The lungs also act as a nice little soft bit of cushion for the lungs or for the heart. The heart moves a lot, and so it's nice to have kind of this packing material, so to speak, of the lungs to accommodate those movements. All right, a little bit here about some basic anatomy of the heart and how it sends blood around the body. Here we have a picture of a heart. And the first thing I like to kind of point out is that the heart really has two sides to it. And you could probably, if you wanted to draw a little bit of a figure here to illustrate where those sides are. Roughly the sides are kind of right here where I've drawn this dotted line. There's a right side of the heart and a left side of the heart. And because we're actually looking at a cross-section of the front view of the heart, keep in mind that the left side will actually appear to be on your right. So this is actually the left side of the heart, even though it's on the right side of the screen. And then this is the right side of the heart, even though it's on the left side of the screen. That would be different if we were looking at the back of the heart. Okay, so let me walk you through this tangled mess of tubes and chambers and things, and hopefully it'll make some more sense for you. There are four chambers to the heart. There are two what are called atria. So this would be the left atrium. This over here would be the right atrium. Atrium is singular, atria is plural. Then there are two what are called ventricles. So the ventricles are these bigger chambers down here. So there's the left ventricle, and there is the right ventricle. The word ventricle comes from the Latin for word for like belly or the depth of something. So this is really the part of the heart that's doing a lot of the work. It's the belly of the heart. And basically, it's on the surface really simple, but then it gets mind-numbingly complex here in a matter of seconds. So hang with me. On the surface, though, it's very simple. The left side of the heart will send blood to what I call, quote unquote, the body. The right side of the heart will send blood to the lungs. And by body, quote unquote, I mean every part of the body that receives blood except for the lungs. Okay. So how this works out is blood always enters the atria. The atria are receiving chambers. They receive the blood, and while they don't really send blood throughout the body like the ventricles do, what their job is they're kind of considered a priming pump. They receive the blood, and then what they do is they contract and they force that blood into the ventricles. They try and pack those ventricles as full as they possibly can, and that's going to greatly increase the efficiency of the heart. All right. We sometimes call the ventricles the working pumps because they're the ones that actually send the blood out of the heart, and then we call these the priming pumps. Okay, so blood will come into the left atrium, into the left ventricle. When the left ventricle squeezes, it will actually shut this valve. We'll talk about the valves in a minute. And blood will be ejected out this big tube right here, which is called the aorta. So we could go ahead and label this. The aorta is one of the great, what we call the great vessels There's several great vessels. Those are the vessels that enter and leave the heart, and they're some of the biggest vessels. Okay, so blood comes around the aorta and it leaves right here. This is going to go to mostly the body. Okay, so goes to all the different places in your body, delivering blood to all of those trillions and trillions of cells that need it. The only exception is the cells that are found in the lungs. That's through a different circuit. All right, here's a question. If the blood that's being sent to the body, or if blood is being sent to the body, do you suppose that it's oxygen rich or oxygen poor blood? I'll let you think about that for a minute. Would it make sense for the heart to send oxygen poor blood to the body? Probably not, because those cells need oxygen. So the blood that's being sent to the body is actually oxygen rich. And because of its oxygen rich, it's actually going to be a red color. So that's a helpful hint to you that anytime you're looking at a heart model and the vessels, leading into the chambers or exiting the chamber appear red, you can be fairly certain that you're looking at something that's entering or leaving the left side of the heart. So you would know, that it's the left atrium and the left ventricle, and then you would also know the names of these valves. Okay, the blood that then goes to the body and then delivers the oxygen is now coming back to the heart, and now it is oxygen poor. And it makes sense that this would go straight to the lungs to go get oxygen, but there's not quite enough power in the heart to send blood to all of the body and then to the lungs afterward, because we lose a lot of pressure as the blood goes and disperses through our body. So there's not enough pressure for that blood to get back to the lungs. So we actually have to send that blood not straight to the lungs. We have to send it back to the heart to kind of get re-energized, regain its pressure, and then continue on its leg to the journey. So the blood actually will come back, and it returns through two ports into the heart. It will return into this vessel and this vessel, both of which will pour that blood into the right atrium. This vessel is called the superior vena cava, and this vessel is called the inferior vena cava. And together, the superior vena cava and the inferior vena cava, you could call them the vena cavae for plural. They pour their blood into the right atrium. It's oxygen poor blood. Okay, blood will then pool into the right atrium. The right atrium will contract. It will push it into the right ventricle. The right ventricle will contract, and now it will send that blood out what is known as the pulmonary trunk. Another great vessel. That word pulmonary refers to lungs. For example, like in Spanish, the name for lungs is pulmones, and so this is anything dealing with the lungs. This is sending blood to the lungs. All right. So we'll have blood going to one lung. So this would be the right lung. We'll have blood going to the left lung. From there, it will receive oxygen. Okay. Then from the lungs, that blood will be sent back to the heart, and it goes through these vessels here and these vessels here. These are called the pulmonary veins. And there's 4 of them, 2 on the left side and 2 on the right side. So you're kind of seeing the story here. Out of the pulmonary trunk, it splits. And actually, I should mention, after the pulmonary trunk splits, it splits into the pulmonary arteries, the left and right. And each one of those arteries will send blood to each lung. So the left pulmonary artery sends it to the left lung. The right pulmonary artery sends it to the right lung. And then it will get oxygen at the lungs, and then it will send it back to the pulmonary veins into the left atrium. Now, what do you think? The blood that's being sent from the lungs back to the heart, what do you think? Oxygen rich or oxygen poor? This would be oxygen rich And you can kind of see that this forms a kind of a figure 8 loop. Blood comes into the left side, goes out to the body, and then it comes back to the right side. Blood then goes out the right side to the lungs, and then back to the left side. We call these circuits, and there is what's called a pulmonary circuit, and there's what's called a systemic circuit. The systemic circuit is the one that goes to the body and back to the heart. Maybe I'll draw this in a different color here. So systemic would be this circuit going from this side of the heart to the body and then back to the right atrium. That's the systemic circuit. And then the pulmonary circuit would be the one that goes from the right side of the heart to the lungs, and then back to the left side of the heart. Sorry, it's kind of messy. A better diagram that kind of shows these circuits is listed in your book, and it's this figure right here. So if you see this figure, this is trying to show you the different circuits. One circuit leaves the left side of the heart and goes to the capillary beds of the upper body and the lower body, and then they send the oxygen, poor blood back, and then from here it goes out to the lungs and then back. So this whole circuit, and that's the systemic. This whole circuit is pulmonary. All right, so that's a little bit about how the heart works. If you really wanted to see a super simple diagram, sometimes this is the diagram that I would draw. So if you're just starting out, it might be helpful to draw this. You go from the left side of the heart to the body, back to the right side of the heart, and then from here to the lungs, and back to this side of the heart. And then these would be your 4 chambers: the left atrium, left ventricle, right atrium, right ventricle. So start off with that diagram just to get an idea of the circuits. Okay, and then once you master that, then you can look at a schematic like this that shows actually how all of the vessels are arranged. Okay, so here we've talked about the chambers and the great vessels and also the 2 circuits. Now let's take it a step forward and let's talk about the valves. There are 2 sets of valves in the heart. There are 2 what are called atrioventricular valves, right here. And they're called atrioventricular. You can abbreviate them AV for atrioventricular. They're called that because they sit in between the atria and the ventricles. Then there are 2 what we call semilunar valves. And the semilunar valves are found actually in the 2 outlets of each side of the heart. So there's one in the right outlet, the pulmonary trunk, the base of the pulmonary trunk. There's one in the left outlet, the base of the aorta. The names of these are actually really simple. The one that's here in the pulmonary trunk is called the pulmonary semilunar valve. The one that's here in the aorta is called the aortic semilunar valve. So those are pretty easy to name. We'll go ahead and label these. And then this one. Okay, now the AV valves are a little bit trickier, less intuitive of a name. But they are, if you're looking at them, you can kind of guess what the name is because they're named based upon how many cusps the valve has. The cusp is kind of like the door or the flaps. In this valve, there are 2 flaps which can close, and so it's called the bicuspid valve. And this one, there are 3 flaps, so it's called the tricuspid. This valve over here is also sometimes called the mitral valve. That name mitral literally means shaped like a mitre, which is the hat that a bishop would wear in the Catholic Church. So if you're Catholic, go ahead and call it a mitral valve, and if you're not Catholic, maybe you'll call it a bicuspid. I'm just kidding. You can use either name. Either name are used quite frequently. Okay, so that's a little bit about the bicuspid valve. I just usually remember these kind of with the mnemonic that you just have to try, right? In this class, you just have to try, right? Okay, so anyways, this kind of somewhat uncertain mnemonic helps you remember that the tricuspid is on the right side. I don't know, it's kind of my silly way of remembering it. Okay, a little bit about these valves. The AV valves, well, in general, what the valve does is it helps the flow of blood in one direction. So, for example, we want blood to come from the atrium into the ventricle, and then from the ventricle out the aorta. We don't want the blood to go into the ventricle and then right back to the atrium, because that would be a very silly and efficient way for blood to circulate through the body, just going back from whence it's come. So the valves help to prevent that. They'll shut off. So after the blood has entered, and then the ventricle now begins to contract and send its blood. The blood has two choices. It can go out the outlet or it can come back in the inlet. But lucky for us, the valve's shut, so it doesn't allow it to leave this hole, and instead is forced out this hole, the outlet. Sometimes the valves get a little leaky, and so this valve doesn't quite close all the way, and so when the ventricle does contract, it will send blood out the aorta, but then a little bit will still seep backwards. That's called mitral valve regurgitation. If it's in the mitral valve, otherwise it would be tricuspid valve regurgitation. But it's also, you can hear the whooshing of blood coming back through that tight opening, and it is going to present as what's known as a heart murmur. So someone could actually hear if this is happening or not. By the way, speaking of talking about heart noises, if you feel in your heart after a big run the ba-bum, 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 ba-bum noise, or if you're listening with a stethoscope, the first bump that you feel is these valves closing. The second bump that you hear is these valves closing. And we'll talk more about this when we get into further detail about the cardiac cycle. But just real briefly, if you were to look kind of at how these valves appear, these are the two AV valves. They're really big. Like this would be kind of the atrium, and then we're looking down into the ventricle, and this would be the other atrium, the left atrium, looking down into the ventricle. And then here's the semilunar valves. I like to point out that the semilunar valves are kind of shaped like a Mercedes-Benz symbol, and the AV valves have these little strings on them called chordae tendineae. The AV valves will open first and allow blood to go from the atria into the ventricle, and then shortly thereafter, when the ventricles contract, it actually forces these valves closed, and then blood will now eject through the two openings of the great vessels, the aorta and the pulmonary trunk. Here's a good view where you can see how there's two cusps, or two flaps to the bicuspid, and there are three cusps to the tricuspid. I don't know exactly what the functional significance of that is or why that is the case, but if you're curious, figure it out and let me know. All right, that's a little bit about the basic anatomy of the heart. Here's just a little review for you to practice. You can go ahead and see if you can label this diagram, and go ahead, pause the video, label it, and then I'll tell you what all the answers here are in a second. Okay, here are the answers for you to check your work. Moving right along, we're going to take a little bit more of a detailed look at the anatomy of the heart now that we've mastered some of the basics. For starters, we're going to look at the layers and the membranes of the heart. If you were to look at a cross-section of the heart wall, you would see several different layers. One, there's what's called the endocardium. Endo means inside heart. The endocardium is a simple layer of epithelial cells lining the heart, and it's actually the endocardium which is going to kind of fold out and then fold back in to form the valves. So valves are made out of endocardium, and it always used to be thought as just kind of a covering, but actually recent research has been showing that there's a whole bunch of things called endothelins released by the cells in this layer that actually help to govern heart development and even circulation. So it's kind of an interesting layer. The next layer is the biggest one. It's the myocardium, and the word myo refers to muscle and cardium refers to heart, so it's basically all of the muscle of the heart, and you can see it's what is mostly the heart when we think of heart, all of the muscle tissue and the way that it contracts and stuff like that. The myocardium is kind of arranged in an interesting fashion. Unlike skeletal muscle, which is just kind of linear, this actually has a lot of swirling texture to it. So there's a band of muscle that wraps around each atria, and then there's a band of muscle that kind of has this figure 8 shape that actually is going to wrap around the ventricles of the heart and squeeze them. So it makes it so that it's a lot more efficient. A squeezing motion is generally a little bit easier than just like pulling something on a linear motion. And so the heart kind of maintains more efficiency when the muscles are arranged in this three-dimensional kind of rotational swirling fashion. All right, here's kind of a look at how the myocardium will present in the different chambers. If you were to look at a cross-section, the big thing that I want to point out here is that compare the difference in muscle thickness between the left side and the right side. Okay, so this is the right ventricle here. Here's the left ventricle. Look at how thin this wall of muscle is right here compared to how thick this wall of muscle is. Each ventricle will eject about the same amount of blood each time it contracts, but the left ventricle has a far more difficult task because it has to send blood to every nook and cranny of the body. The right ventricle just has to send blood to the lungs and back. So you can see why it would make sense. There's far more muscle on the left side. The two ventricles are separated by a wall called the interventricular septum. But you can kind of see that the interventricular septum almost is kind of more one of the walls of the left ventricle than it is really a conjoined wall of the right ventricle. So I like to point this out because if you're looking at a sheep heart, or our models unfortunately don't really show this very well, but if you're looking at a sheep heart on a lab practical, you can always know which side of the heart you're looking at, even if you're not sure if you're looking at the front or the back. You know which side of the heart you're looking at by comparing the outer wall of muscle in each ventricle. Okay, what's kind of interesting is that here's the endocardium, the myocardium. The epicardium is the third layer, and the epicardium is actually going to be part of a layer of the heart, but it's also continuous with part of a sac that surrounds the heart. There is a sac that surrounds the heart called the pericardium. Peri means surrounding and cardium means heart. The pericardium has got 3 layers to it. One is it's made out of the epicardium. So maybe I'll just write a quick note here. This is actually the sac surrounding the heart. So not really technically the heart itself, but it is made up of one layer, the epicardium, which is an actual layer of the heart. So even while the pericardium is not really considered the heart itself, part of its layer is continuous with the actual heart, the epicardium. And then there's also going to be a cavity, so we call that the pericardial cavity. The cavity is any enclosed space in the body. And then lastly, there's the parietal layer of the pericardium. The epicardium would also sometimes be called the visceral layer. And this goes back, echoes to something that we talked about in A&P1, where we talked about serosal membranes. Serosal membranes are a type of membrane in the body that surround organs, and that's the case that we see here surrounding the heart. Just to recall what a serosal membrane looks like, and again, there's one surrounding the heart, there's one surrounding the lungs, there's one surrounding all of the abdominal organs in the abdominal cavity. General feature of a serosal membrane, it's like, you can imagine almost like a squishy water balloon, and this would be the organ here. And then there's kind of 2 walls to the layer, because there's one that's right next to the organ. This is always called the visceral layer of the serosal membrane. And then the outer wall is always called the parietal layer of the serosal membrane. And then there's always the cavity, which contains something known as serous fluid, which is secreted by the cells in these 2 layers. That serous fluid always helps to lubricate the organ so that if it's moving, it's not brushing uh, against other rigid structures. You can think about it being kind of like a little water bed that surrounds each organ. When this type of membrane is surrounding the heart, we call that membrane the pericardium. When this type of membrane is surrounding the lungs, we call it the pleura. When this type of membrane is surrounding the abdominal cavities, we call it the peritoneum. So they have different names, but same feature in each. And then just a quick note to remember that the visceral layer of this is actually the epicardium, the layer of the heart. What's interesting about this serous fluid is that while it's great because it helps to lubricate an organ, and that's especially important on an organ like the heart, which is moving like every beat of every second of every minute of every hour of every day, one of the bad things about it is there's about 15 to 50 mils of fluid in here which can become infected. In fact, there's a famous musician who you might recognize, this guy right here. Anybody recognize this guy? This is none other but the famous Bob Dylan, Minnesota man. And he actually had to cancel one of his shows because he had a fungal infection of this layer. That particular kind of infection was called a histoplasmosis, but that's not something you're going to have to need to know. Just a little interesting bit there. All right, moving on to some other features of the heart. Let's talk about some internal features of the heart, and then we'll talk quickly about some of the external features of the heart. Here's another picture of the heart. This is a good one in your book. We've talked about a lot of the stuff here, but some of the things we've missed are the following. One, the wall in between the ventricles is called the interventricular septum. I guess we kind of talked about that already. Another one is you'll note that on the inner lining of the myocardium, kind of the inward-facing regions of the myocardium, it looks kind of webby. It almost looks like spider webs or something. That texture is called trabeculae carneae. Mind the E here. That's a tough word to spell. And the inside lining is not smooth, and part of the reason for this, it's not super well understood why it's arranged this way, but a good idea is that anytime you have two flat surfaces and you put water between them and you stick them together, they kind of stick to one another. Books will often give the example of putting a glass on a coaster, and when the glass condensation drips down to the bottom of the glass and seeps into between the glass and the coaster actually comes with it when you pull up the glass. So to prevent the walls of the heart from sticking to one another when they contract and they close up, this texture helps to create the space required so that they don't stick. There's also these little strings called the chordae tendineae, and those are attached to papillary muscles. So each one of these is kind of a projection of the myocardium, which is attached to these walls. These are papillary muscles. And I just think this is totally cool. The way that the chordae tendineae and the papillary muscle work is they help to make sure that the valves don't overextend. Okay, so I'm going to have to draw a little drawing here. Imagine this is a flap of a valve and this is a flap of a valve. And this is the open valve, so blood is coming in. Well, when that valve closes, we want the two valves, the cusps of the valve, so let's imagine this is the bicuspid valve and we got two cusps. We want the cusp of those valves to seat perfectly so that blood is blocked from going backwards. If the valves actually closed and then overextended a little bit, now we would have an opening again so that blood can actually seep through the valve. So to prevent that from happening, there are these little cords that are kind of like little guy wires. They grab onto the valves and they prevent them from overextending so that blood could then seep backwards. And furthermore, what's totally interesting about this is that they're attached to these papillary muscles, and the papillary muscles will actually contract to set the tension on these chordae tendineae so that the valves will sit perfectly. And how cool is it that you have these muscles in your heart that can almost do like a physics equation to calculate what kind of tension they need to put on the chordae tendineae in order to close them exactly. If the tension is too loose, they'll flop open. If the tension is not strong enough, they won't hold them right in the right spot. So these little muscles that you never really even thought about that are sitting in your heart right now, every beat of every minute of every hour are putting tension on your chordae tendineae to seal the valves. Now, occasionally this can happen because the chordae tendineae get loose, or something's going on with the papillary muscle, or what have you. And there's actually a procedure where they can go in. They put like a little port into your heart. They go in and they'll actually harpoon like a knot through the valve, and then they'll actually tie it to the papillary muscle and tighten it down to the right tension until it brings the valve back down. That's kind of an interesting procedure. I'm sure if you did a little Googling, I think they call it like the harpoon or something like that. You could go out and watch a video of it. It's pretty fascinating. But in either case, that's the role of the chordae tendineae and the papillary muscles. They help to seat the valves so that they close properly. In essence, they prevent the backwards projection of the valves, or the hyperextension of the valves. There's also this little, in the right atrium, there's this little band called the moderator band, and that's going to help keep the right ventricle somewhat closed as blood pools into here. You know, the right ventricle receives all of the venous blood from the body, so that's a lot of blood coming in. And in order to keep that from distending the right atrium, this band kind of helps. But it also has another sneaky purpose. It helps to take the electrical signals coming down the interventricular wall and kind of shunt them over to this part of the ventricle, so that the ventricle can beat more synchronously, or more unified. We'll talk more about that electrical activity and how it relates to the contraction of the myocardium in another lecture. Okay, there's also a hole here called the fossa ovalis. Actually, I'll back up. When you are in your mother's womb, you are not actually using your lungs because there's no air to breathe inside your mother's womb. So the heart has a sneaky way of getting around this. You know, we don't use the whole pulmonary circuit of our hearts when we're undergoing fetal development in the womb. So there's a hole here which is called the foramen ovale. And that allows blood that's coming, oxygen poor blood that's coming into the right side of the heart to actually jump over to the left side of the heart and bypass the whole pulmonary circuit altogether. It jumps over to the left side of the heart, and then it goes down and it goes out the aorta, and then that will actually lead out to the placenta, which is where the blood will receive oxygen. Once you're born, or probably shortly before you're born, actually, this is going to start to close up. It's either shortly before you're born or shortly after you're born. This will start to close up and seal, and there will still be a remnant of that hole, which is now just called a fossa ovalis. Remember, fossa is kind of like a smooth indentation. Foramen is kind of like a hole. So it was a hole, so we called it foramen ovale, and then it closes up to form an indentation, so we call it the fossa ovalis. All right, some other parts of the heart that we didn't get to look at yet. There's just a couple here. One, continuing on the theme of the circulation in our fetal development, there's another place where blood can get shunted, and that's right here in a region that was called the ductus arteriosum, or ductus arteriosus, I think. That ductus arteriosus, any blood that didn't escape over to the left atrium through the foramen ovalis the foramen ovale will then go out through the pulmonary trunk, and here we have yet another opportunity for blood to shunt over directly from the pulmonary trunk into the aorta. It passed through this little channel, and then after you're born, that channel erodes away, and it just becomes kind of this connective tissue band, which we call the ligamentum arteriosum. The atria, which are these little flaps here on the top, are actually going to have kind of this little structure, this fold that forms the wall of the atrium, and that's called the auricle, because these little folds look almost like a little dog ear, or a sheep ear, or a pig ear. The word auricle literally means ear, and so we call these little folds the auricles. Now the difference between an auricle and an atrium: the atrium would actually be the space, the empty space that's inside here, and then kind of the tissue that's surrounding and encompassing the space. The specific fold here of that tissue right on the outside of the heart here would be called the auricle. So atrium is space, auricle is the actual fold. Okay, this is just a view of the back side of the heart. Not a whole lot to talk about here, other than I just like to point out where you can see kind of what a posterior perspective would look like. You'll see there's the two sets of pulmonary veins bringing blood back into the left atrium. Here's the two sets of pulmonary arteries bringing blood out towards the lungs. Here are the superior and inferior vena cava. Here's the aorta. One thing that I haven't mentioned that's kind of important is the misnomer that veins are always blue and carrying oxygen-rich or oxygen-poor blood, and arteries are always red carrying oxygen-rich blood, because here we see a discrepancy in that convention. Here we see actually an artery. These are the pulmonary arteries carrying. They're blue, so they're carrying oxygen-poor blood, and here we see the veins carrying oxygen-rich blood. So the true convention is this: arteries are vessels that carry blood away from the heart, and veins are vessels that carry blood toward the heart. And 99.9% of the time, this is the case that the arteries are oxygen rich and the veins are oxygen poor. But here's the big exception here: the vessels that carry it out to the lungs are going away from the heart, so they're called arteries. But they are going to the lungs, so they actually have oxygen poor blood, and then vice versa for the veins. So keep this in mind, because this sometimes will trick students up. All right, we're going to talk now about coronary circulation. So the heart, we generally think of it as sending blood to lots of different parts of your body, but the heart has to send blood to itself too, because it is part of your body. So the coronary circulation is kind of like the heart's in-house own little circulatory system, and it's a pretty clever way that it works. Right outside of the aortic semilunar valve, so if I was to kind of draw this continuing in, and maybe this was the aortic semilunar valve, once blood leaves the heart, it will go to the body, and then this valve will open as the blood rushes out, and then once it's kind of pulsed out and then the pressure starts to decrease, this valve shuts again. Well, the heart has to overcome, and we'll talk a lot more about this later, but the heart has to overcome something called the afterload pressure. And that's the pressure in all of the vessels of your body that blood has to move. Anytime you have many feet of tubing that have liquid in it, there's a lot of resistance there. Like if you imagined filling up a garden hose with water, and then it was your job to be on one end of the garden hose and try and blow air through it. I doubt you would be able to blow any air through that hose, because there's so much pressure that you have to overcome that you don't have enough air to push the water through. So the heart has to overcome that pressure every beat. So every time the blood leaves, there's this constant force of pressure pushing back on the heart, which is called the afterload pressure. Well, it's great because right when the blood is leaving the heart, it can overcome the afterload pressure, and it moves and it circulates through the systemic circuit. But then shortly after the ventricle relaxes, there's no longer enough pressure to push that blood through, and so then the afterload pressure starts to push back on the heart. Well, the heart capitalizes on that afterload pressure by using that pressure to actually drive blood flow through what are called the coronary arteries, and the inlets to those coronary arteries sit right above the aortic semilunar valve. So there's always kind of a trick question that can be asked: what is the first set of blood vessels off of the aorta? And a lot of people will think of the ones that are way up here, but in reality, it's these two little small coronary arteries that are right up off of the aortic semilunar valve. These are the first sets of vessels in the aorta. And so after every beat, the afterload pressure will drive blood through those arteries, and it will perfuse the heart and keep it alive. The heart is going to require a lot of energy because it has to beat every second of every minute of every hour of every day, so it requires a lot of blood flow. Now, what's kind of interesting about the coronary circulation is that, or what's interesting rather about the electrical system of the heart, is that the heart actually generates its own electrical pulses. And so I say that because if you are able to keep the heart fed, it can actually remain beating outside of the body. and this is kind of some of the research that I used to do at the U of M. We would take hearts out of mice, and I would make an incision right here in the aorta, and I would put a little kind of needle into the aorta and then suture it onto the needle, and I would actually perfuse fluid, which we call retrograde, meaning it always was continuously being perfused backwards against the normal flow of blood. So if we, hooked the heart up and we started perfusing it continuously retrogradely like this, it would force fluid through the heart and keep it fed. And if you're able to accomplish that, you can actually study the heart and how it beats outside of the body of the animal, which is advantageous because you can actually study the heart function without considering all of the other factors of how the endocrine system or other parts of the animal's body might change heart performance. So it's a good way to study hearts independent of other factors. But in either case, here's a picture of it. This is actually a video that I made. These are my fingers, just giving you an idea of how small these mouse hearts are. So here you can see the needle. It's sutured onto the aorta. What I've done here is I've removed the left atrium, and I'm actually going to insert a little balloon that's deflated through the mitral valve and into the left ventricle of the mouse heart. And it took me a long time to figure out how to do this, you guys, because those hearts are very small, as you can see. I mean, I don't have very big fingers, and that heart makes my fingers look like giant fingers. So right now I'm actually inflating that balloon, and you can see that the heart is beating outside of the body. There's nothing attached to this heart. There's no electrical stimulator or anything. It's beating because I'm feeding it a solution that has sugar in it and also some electrolytes and things that the heart needs. And then I'm going to actually go ahead and now add a stimulator to then get it to beat to a rhythm that's a little bit better suited for our experiment. But nonetheless, then we could inflate that balloon, and actually the balloon was attached to a pressure transducer, so all that to say I could monitor the pressure of that left ventricle and see how well it's beating. That's a procedure known as the Langendorf procedure. There's also other ways to do it where instead of perfusing retrograde, you actually hook up the whole heart so that it's operating really just like a normal heart outside of the body. But all sorts of good stuff there about the coronary circulation. Now, the actual specific vessels of the coronary circulation are the following. The first branches are the right coronary artery and the left coronary artery. The left coronary artery will then also have two branches, one right here, and then this one. This one actually wraps around to the back of the heart, so it's called the circumflex artery. Then there's one that goes down in between the ventricles. Here's about where the right ventricle sits, and this is where the left ventricle sits. So this is called the interventricular artery because it goes in between the two ventricles. It's also called the anterior interventricular artery because there is another that is on the back side as well. Now, this was sometimes called the left anterior descending artery. It was also sometimes called, and this is a little bit more of a grim name, but the widowmaker, because it's actually this coronary artery that often gets blocked in something like a heart attack. Okay, off of the right coronary artery, there are these little branches, which we call marginal arteries. Those are also found off of any of these other main vessels. And then there's a whole bunch of cardiac veins. So what I want you to recall here is that you have a right and left coronary artery. The left continues on to the circumflex and the posterior interventricular. The right just continues on. Off of all of these are these little branches called marginal arteries. There's a whole bunch of veins here. I'm not crazy that you know all the different. I'm not crazy about you knowing about all of the different veins names, but there is one that I'll show you here in a minute. But just know that for every artery, there's also veins draining that blood. On the back side of the heart, then, you can still see the circumflex artery coming back around now to the posterior side. You can also see the right coronary artery coming around to the posterior side. There is going to be those same marginal arteries, those ones coming off of either the circumflex or the right coronary artery. There's also the posterior interventricular artery, so this is the one that's going in between the ventricles, but on the back side now. And then there's a couple different veins and things as well. But I do want to point out that there is this little thing here, which is called the coronary sinus. And this is right on the back side of the heart, right underneath the left atrium. There's this place where all of the venous blood pools into this region called the coronary sinus. And then from the coronary sinus, it will actually deliver that venous blood, which is just returned from the myocardium. It will deliver that back into the right atrium. So here's kind of a drawing of this is showing the right atrium. You can see the fossa ovalis here, the superior vena cava, inferior vena cava. Well, there's a little valve here, a port where all that that venous blood that's just been to the heart can actually rejoin the venous blood from other parts of the body, and then pass its way through the right side of the heart, and then go onward to the lungs. Okay, real quick note here about heart disorders. There's two that we have for you in this particular class to know. One is called angina pectoris. That name literally means choked chest, and it leads to a chest pain from a partial obstruction of coronary blood flow. When we talk about an obstruction of blood flow through the coronary vessels, we call it this word ischemia, and this is actually a word that's used to describe the block of blood flow anywhere in the body, but that also applies here to the heart. And when the heart cells are, when there's ischemia, it means that the oxygen in the blood can't make its way to the tissues of the heart as it should, so it leads to a condition known as hypoxia, which is just a low level of oxygen. When this occurs, the myocardial cells, they will shift from their aerobic respiration, and then they will instead go into an anaerobic respiration, producing lactic acid, which then stimulates the pain. But the big idea here is that this is a partial blockage of a coronary vessel, most likely an artery. Contrast that to what we would call a myocardial infarction. This is what we would think of as a heart attack. That's the scientific name for it. And this is going to be a little bit different in the sense that it is a more of a complete obstruction to blood flow. The difference also between these is that because this is a complete obstruction, here the cells are going to die, and there's actually kind of permanent damage done to the heart. And here the cells do not necessarily die. They just are going to undergo kind of some hypoxic challenge or strain for a while, and then eventually that partial blockage will hopefully relieve itself. When those cells die, it's usually replaced with scar tissue, and the scar tissue, as you can imagine, doesn't really contract as well as it should. Here's kind of an interesting picture that shows these. This is the vessels that have been stained with a dye. And here you can see this would be like the aortic valve, and then you have two coronary vessels coming off of it. This is showing, you can see that this is kind of choked down. There's not as much fluid passing through here because something is stuck in there blocking the vessel. Or you could see there's another blockage here. So this would be one of the things that they could do. They can inject kind of this, I think it's a radioactive dye, or I'm not exactly sure, but something that can be detected through an X-ray. And then they can, see how blood is flowing through these vessels. The reason why these would get blocked is because of like a plaque, like a cholesterol plaque that gets formed in there. So they'll oftentimes, talk about maintaining a proper level of cholesterol to avoid this kind of thing happening. All right, it's high time for some review. So I've got kind of a few questions for you to consider. First, you should be able to describe the path of blood flow through the systemic and pulmonary circuits, and to a level of a degree where you include all of the great vessels, those vessels entering and leaving the heart, the valves that it passes through, and then also the chamber through which it flows. Then this is kind of another big one. Describe the location and function of these following structures that we've mentioned. The endocardium, where is that found and what does it do? Myocardium, where is that found? What does it do? Epicardium, where is that found? Where does it do? Where are each of these found and what do they do? Okay, so think through those. You should be able to explain the mechanism that the heart utilizes to deliver blood to itself, and involving words such as afterload pressure and probably the aortic semilunar valve would be something that you want to talk about. And then have kind of an understanding of where these coronary vessels are located. Like if there was a picture, could you kind of draw where each of these are shown? Again, don't worry too much about all the specific veins, but we do have the coronary sinus here. And then tell me the difference between angina pectoralis or something like a myocardial infarction. All right, our last segment is just a little bit about cardiomyocytes. These are the cells that are found in the myocardium of the heart. They're the ones that are contracting, and they're kind of interesting because they are different than other types of muscle cells. Here's kind of a illustration of what they look like. First and foremost, they have a centralized nucleus, and also you'll note that they're kind of branched, which is interesting for muscle cells. Muscle cells aren't usually branched like this, having many branch points, but this branching that allows kind of that, when we looked earlier, that three-dimensional kind of swirling of muscle around the heart that we saw in one of the first slides of this lecture. Well, because they're so branched, you can get kind of a large network of cells that are kind of interwoven together, so that it's not just a linear bunch of cells all running in one direction. Another thing is that they're quite short. A skeletal muscle fiber can be really, really long, just a super long cylindrical fiber, but these are relatively short. They're branched, they have a central nucleus. They'll have a lot of mitochondria, so in this picture you're seeing all these little mitochondria, and that reflects the fact that they are going to engage in a lot of aerobic respiration. Aerobic respiration is the type of energy production that takes a little bit longer, but it's much more long-lasting. Things like glycolysis or fermentation, they'll make energy really quick, but they won't make very much energy. And they usually make energy out of things like sugars. But aerobic respiration, it takes a little bit longer, but it makes much more ATP, and so it's a lot longer of a process. Somebody like a marathon runner would probably have skeletal muscle cells that have much more mitochondria in them. Somebody that is a bodybuilder, probably not so much. Just a difference in how muscles can utilize energy, either a lot quickly or a little bit, but over a long period of time. Because of this, the heart mostly feeds on triglycerides. At least that's the preferred energy substrate molecule in adults. They also have unique cell adhesions. If we look at the cell-to-cell junctions here, you can see there's kind of this interwoven network here of the two cells. And this presents, if you look under a histological slide, you'll see that there's actually these dark bands that get formed in between the cardiomyocytes, and this isn't really the greatest picture of it, but those are called intercalated discs, and they're a place where the cells are going to be connected to one another. Here's a illustration of intercalated disc. There's this would be like cardiomyocyte A, and then this would be cardiomyocyte B, and this is the gap between them. You can see they're kind of linked by the way that it's interdigitated, or there's like this this pattern that allows a greater surface area for connectivity. And then there are going to be like these little proteins, which books often will describe as being kind of like spot welds. In the same way you would connect two pieces of metal with a little spot weld. It's not like the whole seam is welded, but there's just little points where it's welded together. That's what these are. These are just collections of cell adhesion proteins called desmosomes. They're just proteins that help connect the two cells together. What's also interesting is that there is these things called gap junctions, which are like basically bridges that allow any type of chemical activity or electrochemical activity happening in one cell to then get transferred over to the next cell. So let's say cell number A is being electrically stimulated, and it's generating kind of an action potential or a change in voltage. That the electrolytes that are causing that change in voltage can sweep over to cardiomyocyte B and also cause it to contract. So the heart moves kind of as this one big synchronous unit because of the fact that all of the cardiomyocytes are somewhat tied together electrically. Now there are different places where the electrical signals are insulated from one another so that the chambers can beat independently. We'll talk more about that in a minute, but once we do get to a region or a bundle of myocardium, usually those regions can contract together as a unit. Another thing that's interesting about these is they have a long refractory period. So this kind of goes back to A&P1. The refractory periods, if you don't recall, are the time that it takes for a cell to reset itself before it can contract again. And in something like a skeletal muscle, if we send a nerve impulse to it, well, there's a pretty quick turnaround here where there's the action potential passing along the nerve, and then shortly thereafter there's the contraction. But what's interesting is that by the time the contraction gets underway, it's already kind of past the refractory period. The refractory period is shown here in this graph, and it's like right here. So even before this muscle has contracted, we could actually hit it with another impulse. And the ultimate consequence of that is that it's possible in skeletal muscles to create tetanus, meaning there's such a fast frequency of stimulation that the muscle just stays contracted. It doesn't have like always this nice twitch. That's not how it works in cardiomyocytes. In cardiomyocytes, they have a very long refractory period, so the action potential sweeps along, and then there's kind of this contraction that happens, but the contraction happens the whole time pretty much during the refractory period. Here, the contraction of skeletal muscle happened after the refractory period, but here it happens during. So even while this muscle is contracted, it can almost fully relax before it's ready to be hit with another stimulus and then contracted again. This is important because it allows the heart never to seize. The heart should always kind of twitch with a single contraction. If the heart seizes because the nervous system is hitting it with a bunch of action potentials, then it's not going to pump as it should. The heart needs to beat, and it's those beats that cause the blood to flow. If the heart just contracts and stays contracted, then the blood does not move. So that's kind of interesting. The reason for this is kind of complicated, but it's mostly because of slow calcium channels opening. You know, generally if we're drawing a cell membrane, remember how an action potential works. There's usually more sodium on the outside, and then there's more potassium on the inside. And so the sodium comes in, and that would be the deep polarization. And then the potassium comes out, which would be the repolarization. But in this case, calcium ions, as the cell is trying to repolarize, the calcium ions kind of come back into the cell, and it makes the repolarizing effects of the potassium efflux kind of limited. This is all kind of above and beyond the scope of this class. I do want you to know that there's a long refractory period, and that's due, again, because of these like little leak channels. And the ultimate consequence of that is that the heart can't seize in tetany like any of your other skeletal muscles can. Another thing that's cool about cardiomyocytes is that they can dance. So this is a video of a researcher who has dissolved a heart and plated out each individual cardiomyocyte. That's what these are. And then they're pulsing the fluid, and it's making all of the cardiomyocytes beat to their favorite song. This is another thing I used to do at the U of M. We would take a rat heart out, perfuse it with collagenase that dissolves the extracellular matrix, and then it releases all the individual cells so that you can study how strong they are through the different protocols. That's going to wrap us up for this chapter. So we've got one last review, just describe some of the characteristics unique to cardiomyocytes.